chloroplast cell. Chloroplast have how many parts? What are the differentiation of the cell in based on the mitochondria and chloroplast? Which so this is also here both are this mitochondria DNA, which mitochondria are located in this part. But here is chloroplast, mitochondria versus mitochondria. Ikkadi mitomatis. Here is a chloroplast mitochondria. This outer membrane, inner membrane, cristae. Then cristae. Here is also any granathalagates, stomathalagates inside of the CPDNA, also called a chloroplast DNA, mitochondrial versus chloroplast DNA. But here mitochondria and chloroplast. Important, see the similarities of transfer energies, generative energies, and the mitochondria also got a release of energies. All the chloroplast, double membranes, two same semi-autonomous organelles, move, change, shape, divide. Internal ribosomes, DNA and enzymes, both are similarity, which are called a mitochondria and also got a chloroplast, but here chloroplast structure. The chloroplast organisms is signified by the presence of double membrane. You know that last class is also completed. Both thylakoids and stromal systems are committed for the light reaction and carbon dioxide fixation. That's also respectively. The chloroplast attains a diversified shapes in the different shapes of higher plants, exhibit lens-shaped chloroplast in their cytosol, also round shape, spherical shape, lens shape. The size measure anywhere between 5 and 10 per molecules of length. That is one character of the DNA. This is also chloroplast. But here is also chloroplast double membrane, but here chloroplast structure. But stroma thylakoids and granathylakoids account for the 50% of the total leaf also protein, ribulose, bisphosphate, carboxylase, oxygenases, enzyme, rubisco. That is a major important function which can originated, denoted by chloroplast DNA quality. The chloroplast DNA, which can maintain the rubisco enzymes activity. That rubisco enzymes based to preparation of the food materials in plants by photosynthesis reactions. It's a molecular weight of the 5 lakhs of consists of 8 large subunits and 8 small subunits, and it is created with one of the most abundant available protein in nature. What is a life means? Life is originated from the DNA, but life is continuation depends on the rubisco. Without rubisco formation, do not formation of the food materials by sunlight. That is the rubisco enzymes are most important enzyme which can formation of the nature. Nature means plants, animals, and also inorganic substances, also called a non-living substance. But living organisms, which can classify the two types, the plant's nature, the plant's quality, the plant's development depend on the inorganic substances of the non-living substance. This total rubisco is important. Then it executed photosynthesis by accepting carbon dioxide as the substrate and reduce this carbohydrate status. This one character, but is also this is the chloroplast DNA. Where is the chloroplast DNA located? In the chloroplast. Here is that. Then granathylakoids or stomathylakoids. The stroma means outside of the pillars are plate-like structures. The grana means one above the other. Pillars are which can located that thylakoids inner and outer membrane granum. Then granum also got a stroma. Here is also stroma which can present in the round shape structure of the. Then the several members of the monocots show marginal deviation in their carbon dioxide fixation process, generally known as C4 and C3, also different. Then C3 and C4 means carbon nature, carbon number base to the plants divide C3 and C4. The match, for example, is a C4 branch in which initially carbon dioxide fixation occurs in leaf mesophyll cells containing chloroplast, which lack rubisco and ultimately derived from the starch. That is also major. The enzyme phosphoenol pyrophosphate, phosphoenol pyridase or carboxylase acts as a major enzyme catalyst. First half of the reaction by forming four carbons starting, that is C4, then three carbons starting, but C3, which is then converted into the aspartic acid and malic acid, which are exported to the bundle sheath cell, where they are the decarboxylated and carbon dioxide is reflexed by bundle sheath due to the Rubisco and operate the Kelvin cycle. Then Kelvin cycle means also the number of the carbon dioxide releases. Then the three carbons containing four carbon contains space to dividing the C3 and C4 places. In addition to their role in performing photosynthesis and carbon metabolism, the chloroplasts are involved in the other vital function, such as a synthesis of amino acids and nucleotides, protein synthesis, pigments, and hormones. So these are all is also depends on the Rubisco nature. Rubisco. Then the chloroplat DNA is a comparatively large circular nature, commonly denoted as CT. C small, also call it T small. CT denoted also DNA, not capital. Means which can add to the capital, the DNA quality is not there. DNA act, but DNA before small CT, that is a right name, right written of the ctDNA, also called CP, also ctDNA. The presence of DNA in chloroplast was first identified in 1962. CPDNA, important role. That role is which is responsible for the Rubisco activity. The Rubisco activity depends on the preparation of the food materials in the starch. That is, the size of the chloroplat DNA is usually 140 kilo bases in higher plants and less than 190 kilo bases in lower eukaryotic plants. However, the size of the ctDNA is generally between 120 and 155 kilo bases, not more than 155 kilo bases like a mitochondria. That. So here is the CPDNA is a minimum between 120 and 155 between size of the kilobase of the CPDNA or ctDNA, which are present in the higher plants. By employing DNA binding fluorescent dyes, several copies of the plastid genomes have been visualized. Plastid genome. Plastid means a part of the plastid is which classified there are three types: leucoplast, chromoplast, chloroplast. That is also chloroplast. Then plastid DNA also called chloroplast DNA. Both are the same. The leucoplast and chromoplast does not have DNAs. Only chloroplast has DNA. Means plastid DNA. Also okay. Then chloroplast DNA is also same name. That is, so then the size of the chloroplast genome can comparable to the bacteriophage T4 165 kilobase. There are many copies of circular DNA chloroplast. Example, between 20 and 100 copies per chloroplast in higher plants. That is character. Then in higher plants, the chloroplast DNA exists as a double standard circular molecules. Double standard circular. Double means two helical-like structures, which can closed. That is also round-shaped structure, round organisms. Unlike nuclear DNA, it does not contain 5-methyl cystone or cytosine and is not associated with the stones. It is also biointensity density of around 1.690 gram L by minus 10 to the power of minus 1, which is a corresponding to the guanine and cytosine ratio to approximately 37%. That is a nature. Then measurements is based on the DNA associated through light on the potential coding capacity of the plastome. Then the molecular weight of the plastid DNA is between 80 and 100 millions plastids DNA. Then chloroplast DNA also plastid DNA. Both are, it is also similarity. The weight 100 millions means it is the DNA is between 80 and 100 millions of the plastids. DNA size, which corresponds between 12,000 and 150,000 base pairs of the nucleotides. Adenine, guanine, cytosine, thymine. Adenine, thymine, guanine, cytosine. Always it can take hydrogen and hydrogen bonds. That site is also different, which is also the structure of the chloroplast DNA. So, what are the size of the chloroplast DNA in different plants? Nicotine to vagum 130 kilobases. Then Cucumis sativus 155. Then Citum Pisum sativum 124. Triticum aestivum 135. Germ is 139. Chlamydomonas is also 190. Chlamydomonas means unicellular organisms, which can be important of the algae. That is also chloroplast is present, which is also autotrophic organism. Chlamydomonas 195 kilobases. And the unicellular organisms have more size than then also called a multicellular leg of organism, which is also called a chloroplast genome in a liverwort or unwort mass. The three type of the groups which can classify the bryophytes are first organism plants. That is the chloroplast DNA, which are size in the liverworts. Then the 23S rRNA, tRNA, 23S rRNA. These are all type of the genome. Then also Rubisco is a larger size here. So this Rubisco is responsible to the liverwort autotrophic nature. This autotrophic nature is a first land plants which responsible to the root to stem and leaves or divides in mass plants. Before mass, then liverwort, then RNA-wort. After RNA-worts, complete modified, complete well-developed organism like plants. That is called a polytrichum funaria, which is the first land plants of the nature. Before land plants are evaluated from the Rixia, Markantia, Anthocyrus, and also called Anthocyrus after the polytrichum or funaria. This the size of the rubisco, which are different in the lower organisms like heronivorous, liverworts, and also mass type of the plants. This is also then chloroplast contains one type of the chromosome assume the polyploid natures. It can the DNA replication in a plastids semi-conservative. So in the semi-conservative process, nuclear genome also called a plastid genome, also chloroplast genome, and also called a mitochondrial genome. All is also semi-conservative process. Then, so the semi-conservative in chloroplast of the maize and pea, the DNA replication begins at the two sets about 7,000 base pairs apart, and a procedure or proceeds in the both the directions. Then so one parental DNA, other is also called new DNA threads are formation in the semi-conservative process. Then the several photosynthesis related genes that encoded proteins and located in the thylakoid membranes. So this is also the several evidence confirmed the chloroplast DNA contains the 45 genes coding for RNA and 27 gene coding for proteins. It is a major important. The total 47 genes are coding to the RNA, ribonucleic acids, 27 which are coding the proteins. The total number of genes are located in the chloroplast DNA or plast DNA. These proteins are mainly involved in the chloroplast gene expression. Protein genes are protein and coded parts, and also got RNA coding. Both are responsible for the gene expression. The gene expression is an important role to the metabolic activities in photosynthesis process. The genes coding for proteins of the thylakoids, membranes, and another 10 genes products are committed for electron transport process. A restriction map for maize chloroplast DNA 139 kilobases revealed that Classroom contains unique 22,000 base pairs inverted repeated sequence containing the rRNA genes. That is also one characters. That is also figure, next figure. So this type of the figure here 15.1, 5.1, these are all type of the genes which are located on the then the chloroplast genome or chloroplast DNA. The chloroplast genome, what is the means of the genome means the total genes which are responsible to the photosynthesis activity takes place. That is called a photo and a chloroplast genome. The chloroplast genome is a group on several then a group of the genes responsible for the specific characters. Then all type of the groups are located to the DNAs, which is also called a CPDNA, also called a chloroplast DNA. That is restriction endonuclease map of mage plastid and a mage plastid chromosomes. This is then ribosomes are chloroplast, which have a different in numbers and a presence of 70S ribosomes or 80S ribosomes and 23S ribosomes and 16S Svedberg ribosomes are present. The chloroplast ribosome contain about 50 ribosomal proteins distributed between the two subunits. 23S, 5S, 4.5S. So these are all is also studied in the structure of the chloroplast. But here is also same. But this is also chloroplast or structure. But this is a chloroplast genome. This is also one important of the organisms. Here is also same completed cpDNA, also relative abundant component of the plant DNA. That this is also character features of the chloroplast genome. Characters means major, what are the strictly maternally inherited into the in most angiosperms. Maternally, not paternally here. While in a coniferous inheritance in a paternal. Maternal pad, only coniferous means gymnosperms. Then chloroplast DNA is passed on the from one generation to the next with only any occasional mutations, altering the molecules. Sexual combination does not occur. This is also one kind of molecular system of the cpDNA. So these are the total cpDNA qualities of the all mage. Also other type of the angiospermic glands. This is also molecular systematics on cpDNA. This molecular, which is also restriction enzymes identification, also agrogel electrophoresis are takes place. The gene qualities, the gene nature or DNA structure, which studies in this figure, that molecular systematics of cpDNA. So the entire chloroplast genome involves the isolation of chloroplast DNA from the total DNA. The isolation of the chloroplast DNA from leaf, which can immobilized on agrogel, which can show in the probes, that probes are nature so different in different organism, different type of the plants, the angiosperms, also gymnosperms, that the whole chloroplast genomes of the different brassica species were digested with sac. SDS methods, also got agrogel methods, but molecular systematics of cpDNA, which is also molecular cpDNA, that is also got a nature of the cpDNA. But here is also important bits for preparation of the GL, also got a DL, also got a index of the PG. Then mitochondrial DNA is inherited exclusively from the dash mother, maternally. That is important. The structure of the mitochondrial DNA is dash. Mitochondrial DNA is circular. Chloroplast DNA both are